video, we'll be discussing the adrenal physiology. So we'll talk a little bit about the functional anatomy and histology of the adrenal gland, the hypothalamic pituitary adrenal axis, and we'll go more deeply into the synthesis and secretion of the adrenal hormones, both the steroids and the catecholamines. So in this diagram, we can see the location of the adrenal gland. It is basically a retroperitoneal structure, and it sits above the superior pole of both kidneys. And if we take a cross-section to see the different layers of the adrenal gland, we can see it is divided into two main layers. The first one is the cortex, and then we have the medulla. So the cortex itself, it is divided into three distinct functional layers. So the most superficial one is the zona glomerulosa, which is right underneath the capsule of the adrenal gland, and it basically secretes the aldosterone, which is important in regulating the blood pressure. And then we have the more larger, thicker layer, which is the zona fasciculata. It secretes our cortisol. And then the last layer of the cortex, which is the zona reticularis, and in this layer we secrete the sex hormones of both the males and females. And then lastly we have the medulla, and it secretes our catecholamines, the epinephrine. So this is basically the same thing that we've mentioned in the diagram. One important point to note as a difference between the cortex and the medulla is the embryological origin. And that's why they have distinct functions, even though they're in the same organ. So for instance, the cortex, it is derived from the mesoderm. And going back to our embryology, we know that in the 3rd week of gestation, we have the formation of the trilaminar embryonic disc, which is basically 3 layers. We have the epiblast, and then we have the middle layer, which is the mesoderm, and then we have the last layer, which is the endoderm. And this middle layer, the mesoderm, is the one that gives you the cortex. And then we have the medulla. Its embryological origin is very distinct, as it is originating from the neural crest cells. And those neural crystals would then develop into the chromaffin cells. And those chromaffin cells, they're very distinctive of the medulla, as they have a direct innervation with the sympathetic system. So that's why when you're in the fight or flight response, the sympathetic system gets activated, and it is innervating the medulla. So that's why it stimulates its secretion of both epinephrine and norepinephrine. And that's why we have a lot of epinephrine, norepinephrine circulating in the blood in situations of stress or fear. So here we have the histological slides, and just before going through them, we need to know the cortex, what kind of precursor the cortex uses to synthesize its hormones, and also the medulla, because these precursors, they actually reflect on the histological slides, and you can use them to differentiate between the different layers. So for example, the cortex, it uses cholesterol, and the medulla, it uses amino acids. And you can see here in the different layers of the cortex, you see a lot of whitish droplets, and this is basically the cellular storage of the cholesterol. And you don't actually see it in the medulla here. You don't see as much of whitish deposit of cholesterol, because obviously it does not store it as much because it doesn't need it as the cortex. So going through the layers of the cortex again, we have the zona glomerulosa, the more superficial layer. It comprises 15% of the thickness of the cortex, and we can see the cellular arrangement here is different, because we have the cells in clusters, while in the zona fasciculata we have the cellular arrangement in cords. And the zona fasciculata actually comprises the majority of the thickness of the cortex, 75%. And then the last layer of the cortex, which is the zona reticularis. Here it is not really apparent how the cells are arranged, but it is said that it is arranged in branching cords of cells. So this is that. And then the medulla, here we have the staining with hemotoxylin and eosin staining, the H&E staining. However, if we used a special stain for medulla, we can see the cells that we've discussed before, the chromaffin cells. And these chromaffin cells will appear brownish in color. and then you can see that this is actually the medulla and it contains the chromaffin cells. But here in the cortex, but here in the H&E staining, the medulla appears more of a basophilic bluish color, and the chromaffin is not really apparent here. So as most of endocrine glands, we have an axis that controls it. And here we have the hypothalamic pituitary adrenal axis. So this is axis. And then if we have a situation where we have a drop in the blood glucose or even a trauma that ended with the massive blood loss. So if we took this stimuli, the decreased blood glucose as an example, this would be interpreted by the CNS. The CNS then would stimulate the hypothalamus, relay this stimuli to the hypothalamus, and we know that the hypothalamus is the major relay station of most stimuli in the body. So if we have here the hypothalamus, and this is the pituitary. So this is the anterior pituitary, this is the posterior. The hypothalamus, if it gets this signal, it then would interpret it as we have to increase the blood glucose. And by this, it would increase the corticotroph releasing hormone. If we have the corticotroph releasing hormone from the hypothalamus, it goes to the anterior pituitary, and from the anterior pituitary we would have the secretion of the ACTH to the circulation, and then through the blood ACTH would reach to the adrenal gland, and it will stimulate its growth and also the secretion of its hormones. Also, we can increase the effect of this stimulation by the additive effect of epinephrine. So epinephrine can act as a synergistic hormone to amplify this signal or amplify this increase in blood glucose, because the epinephrine is also one of the stress hormones. So if we take the stress hormones, therefore, one is the cortisol, epinephrine, and also we have the growth hormone and glucagon. And if we already achieved our goal and we increased the blood glucose, so with the adrenal gland already stimulated, it will release cortisol. So cortisol here, it would go and increase the glucose synthesis and also the mobilization of energy storage. So we would increase the glucose. However, if we already achieved this goal, the cortisol would increase in levels. And this increase in levels of cortisol would act as a negative feedback to inhibit the initial stimuli from the CNS and the hypothalamus. So if we inhibit this and we inhibit this, all this pathway would be also cut off, and we would not have as much stimulation as we had before. Just to not forget the other part of this diagram. So if we have, let's say, a decrease in the blood pressure, or we had a major trauma or major blood loss, the hypothalamus, as we said, would interpret this signal, but the hypothalamus then would secrete from the posterior pituitary the vasopressin, also known as ADH hormone, and this hormone would work on retaining more of the of water, so we can increase the blood volume, and from the blood volume we can increase the blood pressure. But this is not exactly the scope of this video, so we actually took the example that is more relevant to the adrenal gland. So now let's get into the synthesis and secretion of the adrenal hormones. So key points to note that all steroid hormones are the result of enzymatic modification of cholesterol. As we said, cholesterol is the main precursor of the synthesis of hormones, main precursor. Steroid hormones are lipophilic because we already synthesized them from a lipophilic molecule, which is cholesterol, and therefore we cannot carry them via the blood freely. We have to attach them carry them by a plasma protease, specifically globulin. And the adrenal cortex produces the greatest variety of steroid hormones than of any other tissue, because, as we said before, the 3 different layers, each layer has a group of cells that is not present in another layer. So for, say, the the zona glomerulosa has the cells that are not present in the zona fasciculata. So zona fasciculata cannot produce aldosterone, for example, and vice versa. So every layer is distinct, and the diversity, and that we have a lot of layers, a lot of cells in one organ, we can assure that a lot of hormones would be produced, a lot of variety of hormones would be produced from this organ. Steroid hormones are synthesized on demand and released immediately. This is a very, very important point, is that we do not store steroid hormones. If we get a ligand signal from here, so this is a signal coming from either the ACTH or whatever signal we have to stimulate this secretion of the steroid hormones. Then we have a second messenger and all that pathway, and then we synthesize from DNA translation, DNA transcription, and so on, and then immediately released to the circulation via carrying it by the globulin plasma protein that we mentioned before. It is not stored unlike other hormones, so it is immediately secreted into the circulation. So in this picture, you can see some of the chemical structures of the steroid hormones. It is not really important to know these structures, but what I wanted to note here is the component or the content of the cholesterol. So as we said, the cholesterol is the main precursor for all steroid hormones, and it is a 27 carbon molecule. And it actually loses some of these carbons in order to give other steroid hormones. So for example, the progesterone is a 21 carbon molecule. So the conversion from cholesterol to progesterone, we lost 6 carbons in this process. Most of the steroid hormones are 21 carbon molecules. So as for example, cortisol and aldosterone. However, we have lost more of these carbons in case of the dehydroepiandrosterone and also androstenedione, which are the 19 carbon molecules. And even we lost even more carbons with estradiol, which is the steroid hormone secreted from the female's ovary, and it is basically an 18 carbon molecule. So this is just a brief introduction about the numbering and how we lost these carbons in the process of the synthesis. Now we can go in more details in how each of these molecules have been produced. So now we know that cholesterol is the main precursor, but how can we obtain it? We can get it from 3 different sources, either from the cholesterol storage of the cell, or we can synthesize new cholesterol in the cell, and we can obtain it from LDL. And LDL basically is low-density lipoproteins, and they're molecules synthesized in the liver. And the liver packs this LDL with cholesterol molecules inside, and then it goes circulating in the blood to deliver this cholesterol into different tissue. And one of these receiving tissues is the adrenal cortex. And LDL is basically a very important source of cholesterol. So once we got our cholesterol, we need to mobilize it from the cytoplasm into the mitochondria. And in order to do this, we need to use a specific transporter that is called STAR. And STAR is basically an abbreviation for steroidiogenic acute regulatory protein. It is a long name, but it is easier to memorize as STAR. And once we got our cholesterol from the cytoplasm to the mitochondrial matrix, we have an enzyme waiting there, which is the desmolase. So cholesterol, and then we have to convert it into another molecule, which is pregnenolone. Okay, and this reaction is mediated by this enzyme, desmolase. So cholesterol, as we saw in the chemical structure in the previous slide, we have here a side chain. And we need to get rid of this side chain because like anything else, any excess part of something you're working on, you need to get rid of it to have a clean cut of this molecule. And this side chain is cleaved by this enzyme. So this enzyme desmolase has another name, which is side chain cleaving enzyme. and it's basically what's the function that is doing. And it cleaves the side chain of cholesterol. So cholesterol is a 27 carbon, and then it gives off this 21 carbon. So basically the side chain is equivalent to 6 carbons, and we already lost this by the function of desmolase. And desmolase is stimulated by ACTH, and this is very important because this reaction is the rate limiting step in the synthesis of steroid hormones, which is the conversion of cholesterol into pregnenolone. So now, after knowing our rate limiting step, we can proceed now with the rest of the pathway. So we already converted cholesterol to pregnenolone, and this pregnenolone is actually the starting point of the whole 3 pathways. So it is easy to think of pregnenolone as a pregnant lady, and she's pregnant with a triplet. And basically this triplet is our 3 main pathways, which will end up with producing aldosterone, cortisol, and androstenedione. So 1, 2, and 3, and those are our triplets. And now let's take each pathway individually. So once we formed our pregnenolone, we can divert this molecule to either of the pathways that we have discussed. So let's say we need to increase our blood pressure, so we need to make more aldosterone, so the body would shift this molecule to the aldosterone pathway. And from pregnenolone, we can then we convert it into a progesterone, and this is by the use of the beta-hydroxysteroid dehydrogenase. It's not really important to know this enzyme, but the most important enzyme that you should know is the 21-hydroxylase, which converts progesterone into 11-deoxycorticosterone. And from this, another important enzyme that you should know is 11-beta-hydroxylase. It converts the 11-deoxycorticosterone into corticosterone. And from there, we end up with our last enzyme, which is a very unique enzyme to this pathway, which is aldosterone synthase. And this aldosterone synthase would convert the corticosterone into aldosterone. And as we said before, that ACTH is the main stimulator for the steroid hormone synthesis. However, in this aldosterone pathway, ACTH has minimum effect on the stimulation, because the main stimulation to secrete aldosterone comes from angiotensin 2. So angiotensin 2 stimulates the aldosterone synthase, and by this we get more aldosterone production and formation. And this is basically it for the aldosterone pathway. So for the second pathway, cortisol formation pathway, of course we have to start with the starting point, pregnenolone. With the use of a 17-alpha-hydroxylase, we get a 17-hydroxypregnenolone. And this 17-hydroxypregnenolone is a shared molecule between 2 pathways, the cortisol synthesis pathway and also the sex hormone synthesis pathway. And if we say, for example, the body is under stress and it needs to increase its cortisol levels, so the pregnenolone will be shifted more towards this pathway, the cortisol formation pathway. So we start from pregnenolone, 17-hydroxypregnenolone, and then we convert it into a 17-hydroxyprogesterone. So be careful, we started here with pregnenolone and then with progesterone, 17-hydroxyprogesterone. Again, we see our very important enzymes again in this pathway, which is the 21-beta-hydroxylase. It converts the 17-hydroxyprogesterone to the 11-deoxycortisol. And this 11-deoxycortisol is modified by the 11-beta-hydroxylase to give off our last end product, which is the cortisol. And one thing to note about the intermediate molecules or the intermediate substances is that they have similar or at least minimum function. So for example, the 11-hydroxycorticosterone and corticosterone, they actually can somewhat increase the blood pressure, but of course not as effective as aldosterone. And also the 11-deoxycortisol have similar function as the cortisol, but they don't actually exert efficient function the same as the main hormones such as cortisol and aldosterone. But they can exhibit some degree of functional activity. And this point is important because once we discuss the deficiencies of these enzymes, you can actually identify which enzyme deficiency this patient has. So, for example, if the patient has no aldosterone activity at all, so you know that there is no intermediate functions, or there's no intermediate hormones that are compensating for this deficiency, so you know that it is a 21 hydroxylase deficiency, and those patients would present dramatically with a severe hypotension. However, if you can see that they present less dramatically, maybe they would not have more severe or they would not have an early presentation. So you know it might not be the 21 beta hydroxylase. It can be the 11 beta hydroxylase deficiency, because the 11 deoxycorticosterone can be compensating for the aldosterone deficiency or aldosterone missing in the body. So for our last pathway, which is the androgen pathway, of course, depending on the needs of the body, we need to synthesize more androgens. Then we will shift the pregnenolone to form more 17-hydroxypregnenolone, and with the use of a new enzyme here, which is the 17-20 lyase. And as the name states, lyase, then we would have some cutting action. So until now, all the intermediates and the hormones that we have discussed are 21 carbon molecules coming from a 27 cholesterol molecule. But now in the androgen pathway, we would reduce this number more. So for from the 17-hydroxypregnenolone is also a 21 carbon molecule. By the 17-20 lyase, we would convert it into a 19 carbon molecule, which is the dehydroepiandrosterone, as we discussed before. And from here, we can proceed with the androgen pathway from the dihydroepiandrosterone into another 19 carbon molecule, which is androstenedione. And this androstenedione is a very weak androgen. And by here, we stop at the adrenal cortex. Whatever continuation of the androgen modification of the hormones would occur either in the testes in case of in the males, or in the ovaries in case of females. So for the testes, in the testes we take this androstenedione, which is a weak androgen. We convert it into testosterone, which is again a 19 carbon molecule. And this is a very strong androgen, but we can convert it into a very potent androgen, which is dihydrotestosterone, which is abbreviated as DHT. And this is by the use of a very important enzyme, which is called the 5-alpha reductase. And this pathway is continued in the testes. However, in the ovaries, so females are very smart. They can convert a 19 carbon molecule into a 18 carbon molecule and actually add an aromatic ring. And this is regarding some of the chemical structures. So basically what you have to know that in the ovaries there is more modification to the androstenedione, which includes the conversion of androstenedione, a 19 carbon molecule, to estradiol, which is an 18 carbon molecule, and it actually has a unique chemical structure, which is an aromatic ring. And this is by the use of a enzyme. So we had already added an aromatic ring, so of course the enzyme would be aromatase. So this is by the enzyme aromatase. Okay. This is basically it. We stop in the adrenal cortex by the formation of androstenedione, and that's it. The continuation depends on the sex of the secondary sexual organs. So, for example, the testes would continue into testosterone and gives off the dihydrotestosterone. However, in the ovaries, we would take the androstenedione, convert it, and take off a carbon molecule. So we end up with a 18 carbon molecule estradiol by the help of aromatase, which also adds an aromatic ring. So this is very special for the females. By this, we finish the pathway of the biosynthesis of the steroid hormones. So the function of the aldosterone is basically to increase the sodium retention, and once we increase the sodium retention, water will follow it, and when we increase the water component, we would increase the blood volume, and by this we will end up an increase in the blood pressure. It also increases the excretion of the potassium and hydrogen out of the body through the kidney. And the function of the glucocorticoids, specifically the cortisol, it increases the glucose levels in the blood by stimulating the gluconeogenesis in the liver. It decreases the glucose utilization by the tissue, and it actually inhibits the tissue from taking up the glucose, thereby increasing more the glucose level in the blood. Except for the brain, of course, because the brain needs to utilize the glucose. Increases the lipolysis, increases protein degradation, and by this we actually increase the precursors to synthesize more of the glucose. We decrease the insulin sensitivity, and actually it has a very important effect, which is the permissive effect to allow the catecholamines, epinephrine and norepinephrine to exert their action. For the androgens in the females, it stimulates the pubic and axillary hair growth and stimulates libido. For men, same as the testosterone, they actually enhance the pubertal growth spurt also. So we're done now for from the adrenal cortex, now going for the adrenal medulla. As we mentioned before, it is derived from the embryonic neural crest cells. It is composed of the post-ganglionic neurons that we already discussed, which is called the chromaffin cells. So the percentage of norepinephrine and epinephrine varies in the circulatory system. So we have more of epinephrine circulating in the blood, and a difference, as we mentioned before, between the steroid hormones that cannot be stored, they are immediately secreted from the cell. Unlike the adrenal medulla, we actually can store it in secretory granules. So we know that the precursor for catecholamine synthesis is amino acid, and in this case it is tyrosine. And we convert tyrosine to L-dopa, which is an inactive form, and from the L-dopa to dopamine. And from dopamine we can convert it into epinephrine. And if we need norepinephrine, we can then further convert epinephrine to norepinephrine. And of course we store epinephrine, norepinephrine to vesicles. And once we get the stimulation from the sympathetic system, that directly innervates the cells of the adrenal medulla, then we can secrete the epinephrine, norepinephrine directly into the circulatory system. And it is important to know that this is the rate limiting step, the conversion of the tyrosine to dopa, and it is by the enzyme tyrosine hydroxylase. And this is mainly the important points in the catecholamine synthesis. So for the function of the catecholamine, you can remember it as fight or flight response. So they would increase the blood pressure, heart rate, and specifically the epinephrine would go and stimulate the bronchioles by bronchodilation, and it would increase the dilation of the pupils, so mydriasis, and decrease the gut motility. So this is the end of this video. Thank you for watching. And these are the references that I've used for this presentation.